Okay, let's unpack this. Have you ever really, truly stopped to think about how incredible your own body is? I mean, how it takes, say, a simple meal
Yeah.
and just transforms it into the actual energy you need to run or think or, well, even just breathe.
It's genuinely amazing. It's like this incredibly complex chemical factory that's running nonstop.
Exactly, a chemical factory.
And today, we're actually going to pull back the curtain on that whole process. This deep dive, it's all about the basic chemistry that makes life tick. We're drawing from Timberlake's Chemistry, an introduction to general organic and biological chemistry.
A foundational text.
Absolutely.
Yeah.
And our goal really is to show you how relevant this stuff is. It's not just abstract science in a book. It's about your health, life sciences,
Yeah.
fundamentally how you work.
And to make it really concrete, we're going to follow a real-world story. It's about Luke, a paramedic, and a health challenge he ran into. His story really grounds these concepts.
It is, yeah.
So, Luke, he's 48, a paramedic, goes for a routine physical. His blood work comes back with a few flags. Cholesterol's a bit high.
Okay.
But the big concern, his liver enzymes, ALT was way up at 282. Normal is like 5 to 35.
Wow, that's high.
And AST was 226, should be below 50. Now, he did mention taking some ibuprofen, some herbs, garlic,
Right.
stuff like that.
Which can affect the liver sometimes. But
Right, but usually not that much. Turns out further tests showed he had chronic hepatitis C. Probably, from needle sticks over his career.
Ah, okay. And that brings us right to the core topic, metabolism.
Exactly.
When you see liver enzymes that high, it's a big sign that the body's chemical factory, especially the liver's metabolic role, is being disrupted. Metabolism at its heart, it's all the chemical reactions happening in your cells,
Right now.
giving you energy, building blocks, everything you need,
So it's happening constantly.
constantly. And you can think of it in two main parts, kind of like breathing in and breathing out. You've got catabolic reactions. These break complex things down. Think food molecules getting dismantled into simpler ones. And importantly, this releases energy.
Like taking apart Legos.
Exactly. And then you have anabolic reactions. These use that released energy to build bigger, complex molecules from the simple parts, like building something new with those Legos, both happening all the time.
Got it. Breakdown and buildup.
Precisely. And that breakdown part, catabolism, usually happens in three main stages. Stage one is digestion, what we just talked about with food. Big molecules, carbs, fats, proteins, broken into small units like simple sugars, fatty acids, amino acids. These get into the bloodstream.
Right. Absorption.
Then stage two. Inside the cells, these small units are broken down even further. A key player here is a little two-carbon piece called the acetyl group.
The acetyl group. Okay.
Yeah. And stage three, this is where the major energy payoff happens, mostly in the mitochondria, the cell's powerhouses. That acetyl group gets oxidized, and that kicks off the process that generates most of your energy currency.
Ah, and that currency, that's ATP, right? This is where it gets really fascinating for me. This universal energy coin.
That's the one, ATP adenosine triphosphate. It's the cell's energy coin. Structurally, it's adenine, a ribose sugar, and crucially, 3 phosphate groups.
3-phosphate.
Yes. And the energy from breaking down food, it gets captured and stored in the chemical bonds holding those last 2 phosphates on. They're often called high-energy bonds.
So the energy is locked in the bond.
Exactly. And when a cell needs to do work, contract a muscle, send a nerve signal, build something, it spends an ATP. It breaks off that last phosphate group.
Hydrolysis.
Right, hydrolysis.
Yeah.
ADP becomes ADP adenosine diphosphate plus a free phosphate. And that releases a neat packet of usable energy, about 7.3 kilocalories per mole.
And then it gets recharged.
Constantly. Those catabolic reactions we talked about, they provide the energy to stick that phosphate back onto ADP, regenerating ATP. It's an incredibly fast cycle.
How fast are we talking?
Get this. In just one day, your body hydrolyzes and regenerates an amount of ATP roughly equal to your entire body weight.
Wait, my whole body weight in ATP, but you don't have that much ATP stored, do you?
Not at all. At any given moment, there's only about a gram of ATP in your cells. It just shows how ridiculously fast that cycle is turning over, recycling constantly.
That's actually mind-blowing. Okay, so if ATP is the cash, how do we get it from, say, lunch? Let's trace it from the food itself. Digestion stage 1. Carbs, fats, proteins. How does the breakdown work?
Let's start with carbohydrates. The breakdown actually starts the second you put food in your mouth. Saliva has enzymes, amylase.
Salivary amylase.
Right. It starts snipping the alpha-glycosidic bonds in starches, breaking them into smaller bits.
Then the stomach.
It pauses there. The acid stops the amylase. But then in the small intestine, it picks up again. Pancreatic amylase continues the job, and then other enzymes right on the intestinal lining like maltase, lactase, sucrase finish it off, producing simple sugars, glucose, galactose, fructose,
And those get absorbed.
directly into the bloodstream.
Wow.
And, this highlights things like lactose intolerance.
Ah, yeah.
If you don't make enough lactase, the enzyme for milk sugar, that lactose travels undigested, causes problems. Knowing the chemistry explains why avoiding dairy or using lactase pills helps.
Makes sense. Okay, what about fats? Triacylglycerols.
Fats are trickier because they don't like water.
Yeah.
So digestion really starts in the small intestine. The key here is bile salts, made by the liver.
Emulsification, right.
Exactly. Bile salts act like detergent, breaking big fat blobs into tiny microscopic droplets called micelles. This gives enzymes, mostly from the pancreas, way more surface area to work on.
Yeah.
They hydrolyze the triacylglycerols into fatty acids and monoacylglycerols. Then something interesting happens. Inside the intestinal cells, they get put back together into triacylglycerols.
Really? Why?
They get packaged for transport, coated with proteins to make them more water-friendly, forming particles called chylomicrons. These travel through the lymph system first, then into the blood, delivering fat to tissues like muscle or adipose tissue for storage.
And fat storage is pretty efficient.
Extremely. Adipose tissue has an almost unlimited capacity, which is great for survival historically, but poses challenges today.
Right. Okay, last one. Proteins.
Protein digestion starts in the stomach. The strong hydrochloric acid there does two things. It denatures the proteins, making them unfold.
Makes them easier to attack.
Precisely. And it activates an enzyme called pepsin, which starts chopping the protein chains. Then, in the small intestine, other powerful proteases like trypsin and chymotrypsin in the pancreas join in and finish the job, breaking proteins all the way down to individual amino acids.
And those amino acids get absorbed.
Yep, into the bloodstream, heading to cells to build new proteins, or if the body needs energy, they can be used for that too.
So all these basic units, simple sugars, fatty acids, amino acids, they're now inside the cells. But they don't just magically release energy, do they? You mentioned partners. Coenzymes.
Absolutely critical partners, especially for the oxidation reduction reactions, the one that really pull the energy out. These coenzymes act like shuttle buses, carrying hydrogen ions and electrons around. Three big ones are NAD plus U, FAD, and coenzyme A.
Okay, NAD plus BSU.
NAD plus stands for nicotinamide adenine dinucleotide. Comes from niacin, a B vitamin. Its job is to accept hydrogen atoms and electrons, becoming NADH. Think of it as picking up energy payload. It's vital in reactions that form carbon-oxygen double bonds, like when the liver processes alcohol.
Got it. NFAD.
FAD flavin, adenine dinucleotide. From riboflavin, vitamin B2, similar job, accepts hydrogens to become FADH2. FAD often steps in when a reaction creates a carbon double bond. You see it a lot in energy pathways.
Okay, NAD plus and FAD handle the hydrogens and electrons. What about coenzyme A?
Coenzyme A, or CoA, is from another B vitamin, pantothenic acid. Its business end is a thiol group. That's a NRGS-H group. Its crucial role is prepping small carbon groups, especially that 2-carbon acetyl group we mentioned earlier, for reactions. It attaches to the acetyl group, forming acetyl-CoA.
Acetyl-CoA. That sounds important.
It's incredibly central. Acetyl-CoA is like a major crossroads in metabolism. Whether you start with carbs, fats, or even some amino acids, they often get converted to acetyl-CoA before entering the main energy-producing cycle. It links everything together.
Okay, so let's follow the energy trail. Take glucose, our main carb fuel. It gets broken down. That's glycolysis, right?
That's the first major pathway for glucose breakdown, glycolysis. It's a sequence of 10 reactions happening right out in the cell's fluid, the cytosol.
Not in the mitochondria yet.
Not yet. Glycolysis itself is anaerobic, doesn't need oxygen. It takes one 6-carbon glucose molecule and splits it into 2 molecules of a 3-carbon compound called pyruvate.
2 pyruvates from 1 glucose.
Correct. And along the way, there's a bit of an energy investment phase, uses 2 ATPs, but then there's an energy payoff phase that generates 4 ATPs. So the net gain from glycolysis is 2 ATP and also 2 NADH molecules per glucose.
2 ATP isn't a huge amount, but it's something.
It's quick energy, crucial for the brain, red blood cells, and muscles during intense bursts when oxygen might be low.
So what happens to that pyruvate next? Does it depend on oxygen?
Absolutely. If oxygen is available, aerobic conditions, pyruvate gets shuttled from the cytosol into the mitochondria. Inside, it's converted into that key molecule, acetyl-CoA. This step releases some carbon dioxide and makes more NADH.
And if there's no oxygen, like in a really hard exercise?
Right, anaerobic conditions.
Right.
Pyruvate stays in the cytosol, it gets converted into lactate, or lactic acid. This might seem like a dead end, but it's vital because the reaction regenerates NAD+ from NADH.
Ah, and NAD+ is needed for glycolysis to continue.
Exactly. So converting pyruvate to lactate allows glycolysis to keep churning out that small amount of ATP even without oxygen. It buys the cell time. That lactate buildup is linked to muscle fatigue and soreness.
Okay, but ideally we have oxygen. Pyruvate goes into the mitochondria, becomes acetyl-CoA. Now what? Where's the big energy payoff?
Now we enter the citric acid cycle,
Mm.
also called the Krebs cycle or TCA cycle.
Yeah.
This happens deep inside the mitochondria.
The powerhouse.
Indeed. The main function here is to take that 2-carbon acetyl group from acetyl-CoA and completely oxidize it to carbon dioxide.
How does it start?
The cycle kicks off when acetyl-CoA, 2 carbons, joins with a 4-carbon molecule already present in the cycle, called oxaloacetate. They combine to form a 6-carbon molecule, citrate, hence the name, citric acid cycle.
And then the cycle turns.
It turns. Through a series of steps, that citrate molecule is rearranged and oxidized. 2 carbons are released as CO2. Along the way, the cycle generates 1 ATP molecule directly via ATP. But more importantly, it loads up more coenzymes. 3 NADH and 1 FADH2 per turn.
And
Critically, it regenerates that starting molecule, oxaloacetate. So the cycle is ready to accept another acetyl-CoA and go again. It's a continuous loop as long as acetyl-CoA is supplied.
So the citric acid cycle produces some ATP directly, but mostly it generates a load of NADH and FADH2. Where do they go? This isn't the big ATP yield yet, is it?
Not yet. This is where the final stage comes in, electron transport and oxidative phosphorylation. This is the main event for ATP production.
And this is also in the mitochondria.
Yes, specifically along the highly folded inner mitochondrial membrane. Think of it like the inner walls have all this machinery embedded in them.
What machinery?
It's a series of protein complexes, numbered I through IV, plus some mobile electron carriers. NADH and FADH2 arrive and drop off their cargo, those high-energy electrons and hydrogen ions, protons.
Like a bucket brigade.
Sort of, yeah. The electrons get passed down the chain from one complex to the next, releasing energy at each step. That released energy is used to actively pump protons from the mitochondrial matrix, the inner space, across the inner membrane into the space between the inner and outer membranes.
So you build up a concentration of protons outside.
Exactly. A steep electrochemical gradient. Lots of protons crammed into that inner membrane space, wanting to get back into the matrix.
And how do they get back?
Through a specialized protein channel called ATP synthase. This is the amazing part. As protons flow back down their gradient through ATP synthase, it causes part of the enzyme to literally spin.
Like a tiny turbine.
Just like a microscopic turbine. And that spinning motion provides the mechanical energy to smash ADP and inorganic phosphate, pi, together, synthesizing ATP. Lots of it. This coupling of electron transport, the chain, to ATP synthesis via the proton gradient is oxidative phosphorylation.
And what happens to the electrons at the very end of the chain?
They need a final acceptor. And that final acceptor is oxygen. Oxygen picks up the electrons and combines with protons to form water, H2O.
Ah, so that's why we need to breathe oxygen. Without it.
Without oxygen, the whole electron transport chain backs up.
Mm.
NADH and FADH2 can't unload their electrons, ATP synthesis grinds to a halt, and the cell runs out of energy quickly. That's why aerobic respiration is so efficient, but absolutely requires oxygen.
How much ATP are we talking here?
On average, each NADH that enters the chain yields about 2.5 ATP. Each FADH2 yields about 1.5 ATP.
Okay. And there's a health link here too, right? It's something about uncoupling.
Yes, interesting point. Certain substances, called uncouplers, can disrupt this process. They basically poke holes in the inner mitochondrial membrane, allowing protons to leak back into the matrix without going through ATP synthase.
So the energy from electron transport is still released.
But instead of making ATP, it's just dissipated as heat. This isn't always bad, though. Brown fat, which babies have a lot of, uses a natural uncoupling protein precisely for this purpose to generate heat and keep them warm. It's called thermogenesis.
Fascinating. So if we tally everything up, from one molecule of glucose going through glycolysis, pyruvate to acetyl-CoA, the citric acid cycle, and finally electron transport, what's the grand total ATP count?
Adding up the ATP made directly and the ATP from all the NADH and FADH2 generated along the way, the complete oxidation of one glucose molecule yields a grand total of about 32 ATP molecules.
32 ATP from one glucose. That's way more than the 2 from glycolysis alone.
Immensely more. It shows the power of aerobic respiration and those mitochondrial pathways.
Okay, but we don't just run on glucose. What about when carbs are low? Our bodies are adaptable. You mentioned fat.
Extremely adaptable. Fatty acid oxidation is a huge energy source, especially during rest or prolonged exercise. This also happens primarily in the mitochondria.
How does it work? Is it similar to glucose breakdown?
It's a different pathway called beta oxidation. Basically, the long fatty acid chain gets chopped up, 2 carbons at a time, from the carboxyl end.
2 carbons. That sounds familiar.
It is. Each 2-carbon chunk that gets cleaved off forms 1 molecule of acetyl-CoA.
Which can then enter the citric acid cycle.
Exactly. Plus, each round of beta oxidation also produces 1 NADH and 1 FADH2, which head off to electron transport. This cycle repeats, chopping off 2 carbons at a time, until the whole fatty acid chain is converted into acetyl-CoA molecules.
And since fatty acids are often really long chains,
They produce a ton of acetyl-CoA, NADH, and FADH2. A typical 16-carbon fatty acid, for example, can generate over 100 ATP molecules. That's why fat is such a dense energy store.
which, as you said, is biologically advantageous for survival. Think hibernation, migration.
Right, camel's humps, whale blubber, it's stored energy and insulation. But in humans today, with abundant food, that efficient storage can lead to obesity and related health issues like diabetes, heart disease.
Right. Is there hormonal control?
Yes, a key hormone is leptin, produced by fat cells themselves. It signals the brain about how much fat storage there is, helping regulate appetite and energy expenditure. It's complex, though.
Now, what happens if someone is breaking down a lot of fat very quickly, maybe on a very low-carb diet or in uncontrolled diabetes? Does the system get overloaded?
It can, yes. If massive amounts of fatty acids are being oxidized, the liver produces a flood of acetyl-CoA. If the citric acid cycle can't keep up with processing it all,
Which might happen if carbs are low because oxalacetate for the cycle can be derived from carbs.
good point. So this excess acetyl-CoA in the liver gets converted into alternative fuel molecules called ketone bodies. The main ones are acetoacetate and beta-hydroxybutyrate, plus a little acetone. This process is ketogenesis.
And these ketone bodies can be used elsewhere.
Yes, the liver releases them into the blood, and tissues like the heart, brain, and muscles can take them up and convert them back into acetyl-CoA to use for energy. It's an important backup fuel system, especially for the brain when glucose is scarce.
But too much is bad. That's ketosis.
Moderate ketosis, like on a ketogenic diet, is usually manageable. But if ketone body production drastically outpaces their use, they build up in the blood. Since 2 of them are acidic, this leads to ketoacidosis, a dangerous drop in blood pH.
And this is a major risk in.
Uncontrolled type 1 diabetes, primarily. Without insulin, glucose can't get into cells, so the body relies heavily on fat breakdown, leading to runaway ketogenesis. Can also happen in severe starvation or alcoholism. That fruity acetone smell in the breath is a classic sign.
Okay, so carbs and fats are the main fuels. What about proteins? When do they get used for energy?
Proteins are primarily for building blocks, amino acids for making new proteins, enzymes, hormones, etc. They only contribute significantly to energy needs, maybe 10% under normal conditions. But during prolonged fasting or starvation, when carb and fat stores are depleted, protein breakdown ramps up.
How are amino acids processed for energy? It seems more complex because of the nitrogen.
It is. The first step, mainly in the liver, is usually to remove the amino group, NH2. This often happens via transamination, swapping the amino group onto another molecule. Then oxidative deamination removes the amino group from glutamate, releasing it as an ammonium ion, NH4+.
And ammonium is toxic.
Yes, highly toxic if it builds up. So the liver has another crucial pathway, the urea cycle. This cycle takes those toxic ammonium ions and converts them into urea, which is much less toxic and can be safely excreted by the kidneys in urine.
That's why kidney function is linked to urea levels, like BUN.
Exactly. Blood urea nitrogen, BUN, is a measure of urea in the blood, reflecting both protein breakdown and kidney function. Once the nitrogen is removed, the remaining carbon skeletons of the amino acids can be converted into intermediates that feed into glycolysis or the citric acid cycle, contributing to ATP production.
Wow. The body really has backup plans for its backup plans.
It's incredibly resourceful.
Okay, let's circle back to Luke, our paramedic with hepatitis C. His initial treatment didn't quite normalize his liver enzymes. What happened next?
Right, his ALT and AST were still elevated, indicating ongoing liver inflammation or damage, despite some initial therapy. So his physician moved to a more potent combination, antiviral drugs, specifically interferon and ribavirin.
How do those work?
They work together to fight the virus directly. Interferon boosts the body's own immune response against the virus, while ribavirin interferes with the virus's ability to replicate its genetic material. They attack the virus's life cycle.
And the outcome for Luke?
It was much better. After about 8 weeks on this combination therapy, his liver enzymes showed significant improvement. And after 4 months, both his ALT and AST levels were back within the normal range.
That's great news.
It is. Of course, he'll need lifelong monitoring as Hep C can be persistent. But it really shows how targeting these specific biochemical pathways, in this case, viral replication, can directly impact organ function and those metabolic markers like liver enzymes. Understanding the chemistry leads to effective treatments.
What an amazing journey we've covered. From, just eating food, through digestion, into the cell, glycolysis, acetyl-CoA, the citric acid cycle, that whole electron transport chain.
Well,
All powered by these coenzymes and ultimately making ATP.
it really is a symphony, isn't it? And seeing how it all connects carbs, fats, proteins, energy production, health conditions like Luke's or diabetes, or even just athletic performance, it highlights why understanding this chemistry is so fundamental. It's not just abstract, it's the chemistry of us.
The body's ability to manage energy, switch fuels, keep everything running, it's just brilliant.
Absolutely brilliant.
So maybe a final thought for everyone listening. Next time you eat something, or even just, sit and think, maybe take a second to consider that incredible, intricate dance of molecules happening inside you, constantly fueling every single thing you do. What tiny part of this whole chemical factory do you find most amazing now?
It definitely gives you a new appreciation for the mundane act of eating or breathing.
It really does. From all of us here at the Deep Dive, and especially the Last Minute Lecture team, thank you so much for joining us on this fascinating journey into the chemistry of life.
We hope this deep dive has helped you feel truly well informed.